Hello everyone. So today I'm going to present about the combination effect of lapatinib and palbociclib in endocrine resistant breast cancer cells. My name is Kantasorn Hopattapon and my advisor is Associate Professor Vanarasamy Getchat. The three most common cancers worldwide are colon, lung, and breast cancer in both sexes. Particularly in women, breast cancer is the leading malignancy and occurs most frequently by 1 in 8 to 10 women. However, a decline in mortality rate is seen in North America and Europe due to early detection by mammograms and ultrasound and new emerging effective systemic therapies. On the contrary, in Asia, Africa, and South America, there is an increasing trend in breast cancer incidence, probably caused by lifestyle changes and more developed screening programs in developing countries. Moreover, the mortality rate in these areas is rising. So, estrogen plays an important role in breast cancer. Estrogen are sex hormones produced from tissues inside our body, and then it binds with estrogen receptors in the cancer cells and promotes cell growth. Types of breast cancer can be categorized into histological subtypes and molecular subtypes. Today we'll be focusing on the molecular subtypes, which is categorized into 4 molecular subtypes based on their cancer gene expressions: luminal A, luminal B, HER2 enriched, and basal-like or triple negative. These subtypes are separated by the expressions of estrogen receptor, progesterone receptor, and HER2. Ki67 also plays an important role in investigating how aggressive the breast cancer can proliferate. Surgery and radiotherapy or local therapy are the first choices for patients with early-stage breast cancer. Apart from local therapy, systemic therapy still plays an important role in almost all breast cancer patients, including early and advanced stage cancer patients. So, as mentioned before, there are around 4 subtypes, and estrogen receptor and progesterone receptor positive tumors can be treated by endocrine or hormonal therapy. HER2 positive tumors can be treated by targeted therapy, or the most well-known one is trastuzumab. And the triple negative subtypes can be treated by chemotherapy. And this is the figure shown about its treatment and its subtypes. And for endocrine or hormone therapy, there are 3 main anti-estrogens. The first one is selective estrogen receptor modulators, or SERMs. SERMs blocks estrogen from binding to estrogen receptor. Secondly is SERDs, or selective estrogen receptor downregulators. SERDs blocks estrogen from binding to ER and helps degrade ER. Aromatase inhibitors, or AIs. AIs blocks production of estrogen from tissues. So, SERMs are anti-estrogens that compete with estrogen and have a different effect on different tissues. For example, tamoxifen inhibits proliferation of breast tissue, but it is an agonist for the uterus, the heart, and the bone. SERDs, or the most known SERDs, fulvestrant, are anti-estrogens that induce ER degradation and demolish the ER signaling pathway. Therefore, it acts as a universal estrogen receptor antagonist, and it is effective on tamoxifen-resistant breast cancer. Estrogen receptor and progesterone receptor levels were reduced more than tamoxifen if treated with fulvestrant. There are many mechanisms that can trigger endocrine resistance in breast cancer patients. The first one is metabolic resistance. Typically, tamoxifen will be converted to its active metabolites by CYP enzymes in the liver. The main responsible gene for this is the CYP2D6 gene, and 7% of patients are poor metabolizer of tamoxifen. Low levels of active metabolites resulted in worse prognosis. And as mentioned before, tamoxifen acts as an ER antagonist in breast tissue, but acts as an ER agonist in bone, uterus, and the cardiovascular system. These variations could be from diversity in the expression of co-regulatory proteins in the cells. The most known coactivator is the nuclear receptor coactivator 3, or NCOA3. Patients with high level of NCOA3 had resistance with tamoxifen and also chemotherapy, which leads to worse prognosis. Another mechanism is HER2 and PI3K AKT-mTOR pathway. HER2 activation in endocrine-resistant breast cancer cells activates PI3K, AKT, and MAPK-ERK pathways. The signaling pathways promote cell proliferation and survival. PI3K mTOR pathway also correlates with resistance to hormonal therapy by activating estrogen receptor in the absence of estrogen. So PI3K activation lowers estrogen receptor expression and resulted in tamoxifen resistance. This signaling pathway promotes self-proliferation and survival. So in MTF7, LTC2, and LTC9, which are tamoxifen resistant and tamoxifen and fulvestrant resistant cell lines, have more HER2 pathway and its downstream signaling proteins such as PAKT and PERK12. These proteins were increased compared to MCF7 wild-type cell lines, and the levels were similar to SKBR3 or the HER2 overexpressed cell line. RB or retinoblastoma is a tumor suppressor protein, and it binds with E2F to prevent gene transcription process. However, CDK4/6 can bind to cyclin D1 and together hyperphosphorylated retinoblastoma and lead to cell cycle progression. This process contributed to the resistance to anti-estrogens. Fortunately, palbociclib, a CDK4/6 inhibitor, is developed and can be used in combination with fulvestrant to treat ER-positive breast cancer patients. So we have talked about endocrine-resistant mechanisms, and now we're going to move to the hallmarks of cancer, which will be focusing on the metastasis and the EMT pathway. So metastasis starts with epithelial mesenchymal transition, or the EMT pathway, and then local tissue invasion, or extracellular matrix degradation, and thirdly, intravasation to the blood vessels, and then homing in the vessels, and then extravasation to other tissues, and then metastatic niche formation. So, epithelial mesenchymal transition, or EMT, is a cellular process important in metastasis. Cells lose their epithelial phenotypes and gain more mesenchymal phenotypes. The monolayer sheath cells initiate mobilization through EMT. This process is associated with downregulations of epithelial cell markers such as E-cadherins and occludins, and upregulations of mesenchymal markers such as vimentin and N-cadherin. Transcription factors like SNEL, TWIST, and ZEB12 are also known as regulators of the process. Finally, EMT leads to invasion, migration, metabolic reprogramming, and apoptotic resistance. The process of breast cancer metastasis starts from the cells overexpress pro-metastatic proteins, including CXCR4, CXCR2, and MMP9. These proteins together allows metastatic cells to invade into the matrix, extracellular matrix, and then to the bloodstream. Chemokines in the bloodstream or surrounding tissues direct the migrating cells by signaling through their receptors. Cells that have successfully migrated to the surrounding tissues may undergo angiogenesis through vascular endothelial growth factor, or VEGF, and epidermal growth factor receptor, or EGFR, and develop new distal tumor in the invaded tissues. MMP9 is also known together with CD44 and TGF-beta, which increase proliferation of the metastatic cells. Wnt-beta-catenin pathway is a conserved pathway through evolution and controls the EMT. The Wnt pathway gene expression also upregulates cell proliferation, invasion, and metastasis, and also angiogenesis. Furthermore, endocrine-resistant breast cancer cells also exhibit overexpression of Wnt-responsive genes. Moreover, higher metastasis of breast cancer to lung and brain is higher if the patients are identified by the Wnt-beta-catenin classifier. So we have talked about the breast cancer and its mechanism of how it has been resistant to endocrine therapy. Now we're going to talk about the drugs. Palbociclib. Palbociclib is an orally selective CDK4/6 inhibitor used in combination with fulvestrant or aromatase inhibitor to treat ER positive with HER2 negative, which are endocrine treatment resistance. Its side effect is myelotoxicity, which can be reduced by dose reductions. Palbociclib decreases retinoblastoma and TSC2 phosphorylation and attenuates mTORC1 activity, which then has a feedback inhibition of upstream EGFR family kinases and delayed tumor recurrence in HER2 positive breast cancer in transgenic mouse models. Next is lapatinib. Lapatinib is an oral dual tyrosine kinase inhibitor which blocks HER1 and HER2 at the intracellular domain and resulted in tumor cell growth suppression. In cutaneous squamous cell carcinoma, lapatinib enhanced apoptosis and interrupted PI3K-AKT-mTOR pathway and decreased the Wnt-ERK PI3K-AKT pathway. Its side effect is diarrhea, which is well tolerated, and lapatinib is approved for hormone receptor and HER2 positive breast cancer combined with letrozole. So, palbociclib plus fulvestrant is used in endocrine system breast cancer patients nowadays, and estrogen receptor and HER2 signaling pathway, they interact with each other through many pathways. And in LCC2 and LCC9 cell lines, HER2 and its downstream signaling proteins were increased. Thus, adding lapatinib could potentially result in tumor cell growth suppression. In HPV-negative head and neck squamous cell carcinoma, simultaneous inhibition of retinoblastoma 1's phosphorylation with palbociclib and of EGFR activity with lapatinib resulted in synergistic inhibitory effects on the squamous cell carcinoma proliferation and suppressed ERK1/2 phosphorylation. Clinical use of HER2-targeted drugs in endocrine system breast cancer patients whose tumor overexpress HER2 has not been included yet in any clinical guidelines, so this is the rationale of this study. So, the combination of lapatinib and palbociclib could potentially inhibit cyclin D1, NCOA3, and HER2 signaling pathway, and inhibit the EMT process by Wnt beta-catenin pathway in MTF-62 and LCC9 cell lines. So, this is a research gap that this combination could inhibit the growth of the endocrine system breast cancer cells. And the objective of this study is to study the effect of combining lapatinib and palbociclib in endocrine-resistant breast cancer cells, and to study the molecular mechanism of lapatinib and palbociclib, which includes anti-proliferation, anti-HER2 activity, anti-invasion, and endocrine system breast cancer cells. The questions of this research are: does combining lapatinib and palbociclib show synergistic effect in inhibiting proliferation and migration invasion in endocrine system breast cancer cells? And does combining lapatinib and palbociclib inhibit cyclin D1 and NCOA3, the EMT process by Wnt beta-catenin pathway, and HER2 signaling pathway in the cells? The hypothesis correlates with the questions, and it is: lapatinib combined with palbociclib exhibits synergistic effect in anti-proliferation, anti-migration invasion, and anti-HER2 activity in endocrine system breast cancer cells. And lapatinib combined with palbociclib also inhibits cyclin D1 and NCOA3, the EMT process by Wnt beta-catenin pathway, and AKT HER2 ERK pathway in the breast cancer cells. So the study design of this research is in vitro experimental research. The conception framework is here, and the experimental design is here. So we're going to study about the cytotoxic activity of the cells by cell proliferation by looking at the MTT assay. And for the mechanism, anti-invasion activity, we're going to be looking at the cell invasion by Matrigel assay, and Wnt beta-catenin and EMT markers by Western blot. And for HER2 signaling is also Western blot, and for resistance to tamoxifen, we're going to look at NCOA3 and cyclin D1, which can be investigated by real-time PCR and Western blot. Methodology. So first we're going to look at the MTT assay, or the cell viability assay. So we prepare MCF7, LCC2, and LCC9 cells in minimum essential media, which is added with 5% fetal bovine serum 1% penicillin and streptomycin and amphotericin B. The cells are cultured in T25 flasks and put in the incubator, which is controlled in a 5% carbon dioxide gas and 37 degrees Celsius environment. Media are changed routinely every 2 to 3 days, and when the cell is dense, the cell will be subcultured into a new passage. Preparation of the drugs pure compounds are dissolved in DMSO into a stock solution in 10 millimolar concentration, and the stock solution is kept in minus 20 degrees Celsius. When the drug is going to be used, it will be dissolved in complete media to get the specified concentrations. So for the cell viability assay, or the MTT assay, we'll seed a 96 well culture plate with 5,000 cells per well, left for 24 hours, and then treat with drugs in different concentrations, left for 24, 48, or 72 hours, and then add MTT, thiazole blue triazoline bromide, 10 microliters per well, and left for 4 hours. After that, formazan crystals are added with the MSO, and the plate is then read by a microplate reader at 570 nanometer absorbance. So next we're going to be looking at the RT-PCR, real-time reverse transcriptase polymerase chain reaction. First, the cells are cultured on 6 well plate for 24 hours. Medias are removed and drug solutions are added at a concentration of IC50 and 2 concentrations lower than IC50. Negative control is the MSO, and then we incubate for 48 hours. The RNA is extracted using triazole reagent. Total RNA is reversely transcribed into complementary DNA by the reverse transcription system, and then is used as a template for subsequent real-time PCR amplification. Cyclin D1 and NCOA3 are tested with real-time PCR amplification using specific primer. Next, we're going to talk about Matrigel assay. Matrigel assay is a breast cancer cells invasion functionality test. Upper chambers of the transwell are coated with Matrigel and incubated for 24 hours. 50,000 cells per well are added to upper chambers in serum-free media, while lower chambers contain 5% serum media, so the cells will come down. And then drug solutions are added with different concentrations and then incubated for 48 hours. And then we scrape off the non-invading cells from the upper chamber and fix invasive cells with 4% formaldehyde and stain with crystal violet dye and count the invaded cells under a microscope, and then we can see if the cells invaded or not. Next, we're going to be looking at the Western blot. So Western blot, first we'll prepare the sample by lysing the cells with buffer containing proteinase inhibitor cocktails, and then cell extracts are loaded to acrylamide gel and transferred onto a nitrocellular membrane. Blots are blocked by the blocking buffer, and then we are going to run the gel and incubate, and then transfer the membrane and incubate with primary antibodies for NCOA3, cyclin D1, AKT, HER2, ERG, or Wnt1 or beta-catenin pathway in 5% non-fat dried milk in buffer overnight. And then we wash the membrane twice and incubate with a secondary antibody, and then immunoblots are developed using ECL Western blotting substrate, and the band intensity is then quantified. So that is the end of my seminar presentation, and the question for take-home message is to give an example of endocrine resistant mechanisms in breast cancer cells. Okay, so that's it for today, and thank you everyone for listening, and if there are any questions, feel free to ask. Thank you.